pedigrees to detect short-term selection in natural populations. So as you all know, and as we just heard about in this session, we know that evolution can occur over just a few generations. So classic examples of rapid phenotypic evolution include the evolution of melanism in peppered moth in the UK, as well as evolution of beak size and shape in Darwin's finches. And as we just heard about, we also know that there's rapid evolution in guppies, monarch butterflies, and daphnia. But what about rapid evolutionary dynamics at the genomic level? So can we see evidence of selection shaping short-term allele-frequency dynamics at the genome? So studies of contemporary evolution at the level of the genotype have been pretty rare so far, mostly due to limited temporal sampling of natural populations through time. And some of the best opportunities to study kind of the genetic basis of contemporary evolution come from a handful of long-term demographic studies in which individuals are followed in a population across decades. And so I work on one such system. The system I work on is the Florida scrub jay, Aphelecum corvalescens. A population of these scrub jays has been intensively monitored at Archbold Biological Station since 1969. And by intensively monitored, I mean really intensively monitored. So we have really accurate data on individual lifespans, as well as annual and lifetime reproductive success for thousands of individuals in our population. Nestling an immigrant into our population has been banded and blood sampled. So what this means is that we've accumulated a data set of genomic, life history, and morphological data for more than 4,000 individuals on a 12-generation pedigree. So here I'm showing you the genealogy of one particular individual. So this is just showing all of its ancestors in our population through time. If you look closely, this is kind of fun because this particular individual actually has several inbreeding loops. Our full pedigree looks something like this. So what we've done is design custom Illumina isolate B chips and genotyped about 3,800 individuals in our population at 12,000 SNPs scattered across the genome. So here I'm showing you in gray bars the total number of either adult or juveniles in our population through time, and in blue I'm showing you the number of individuals that we have genotype data for. So as you can see, we've sampled nearly every individual in our population for the past 10 years. So using these data, we can look at allele frequency dynamics at SNPs across the genome. Here I'm showing you allele frequencies. So to measure allele frequencies, I'm using the adult breeding individuals in our population from year to year. I'm showing you trajectories for our 12,000 SNPs, and I've highlighted trajectories for SNPs that show a really large shift in allele frequency in this case, an allele frequency shift of greater than 0.17 between 1990 and 2013. And so what we're interested in now is to understand what are the processes that govern these allele frequency dynamics we observe in our population through time. Does drift explain everything we see, or does gene flow and selection also play a role? So typically, population geneticists in the past have compared observed allele frequency shifts to those you would expect under a Wright-Fisher model. However, we know that our population violates most assumptions of the Wright-Fisher model. We have overlapping generations, and we also know that when we sample our individuals, we have related individuals. Another thing too is that previous work has shown that we actually have really high levels of immigration into our population. So here I'm showing you the proportion of new breeding individuals that are new immigrants, so a measure of immigration rate into our population over time, and you can see that it ranges from 0.15 to over 50%. So given this high level of immigration and the problem of overlapping generations, related individuals, and whatnot, how should we think about allele frequency changes through time? The approach we use is this method called gene cropping. So for every single population, there's a true underlying pedigree, which constrains how genetic drift can act in population, you have a lot more power to distinguish selection from drift. In this case, we're actually going to use gene dropping simulations to estimate the expected genetic contribution of immigrants to our population through time. And the way we do this is we start out with our population. For the rest of this talk, I'm going to start our simulations in 1990. So the breeding birds present in our population are the founders, and for this particular simulation, we assign all of our founders a genotype of 1-1, and then any immigrant that appears in our population in more recent years, so from 1991 onwards, will assign a different genotype. We then simulate Mendelian transmission of alleles down the pedigree, do this a million times, and this will generate kind of expected allele frequencies in our population under neutral processes alone. And so in this particular case, if we're interested in estimating the genetic contribution of immigrants, what we want to look at is the frequency of this immigrant allele from year to year. So this is what I'm showing you here. This is the expected genetic contribution of recent immigrants into our population. So remember that we started our simulations in 1990. Here we're assuming that every bird in our population in 1990 is considered a resident. And we're looking at the expected genetic contributions of immigrants that show up in our populations after 1990. You can see that it increases in, by 2013, more than 75% of observed levels of genetic diversity in our population likely came from immigrants. We can break this pattern down a little bit more by assigning an immigrant appearing in our population in any given year a different allele. We'll just give them a different number. And look at the expected contribution of immigrants from year to year. So here I'm showing you these are stacked lines showing the contribution of immigrants from each year. So for example, in this red line is the expected genetic contribution of immigrants that showed up in 1991. The orange line shows the additional expected contribution of immigrants from 1992 and so forth. So given that we know that immigrants should contribute a lot to our population, we would expect that they would also play a large role in our allele frequency trajectories. And indeed, we do see that immigrants can cause large allele frequency shifts. Here's one example SNP that has increased in frequency through time. We know that the founder allele frequency is 0.44, the immigrant allele frequency is 0.58. And so for our gene dropping simulations, where we're trying to understand the allele frequency dynamics, we use the observed allele frequencies for our founders and for our immigrant individuals. And here in black, the black line I'm showing you the mean value from our gene dropping simulations, and the gray bars indicate the 95% confidence interval. And here our gene dropping simulations nicely show that indeed this large allele frequency shift that we see in this particular SNP is due to immigration into our population. So our gene dropping simulations can account for gene flow and drift. Now, how do we detect for selection after controlling for those two processes? Here I'm just looking for net selection between 1990 and 2013, and the way we do this is we run our million gene dropping simulations to generate the expected allele frequency in 2013 under neutral processes. This is shown in gray. For both SNPs, if the actual observed frequency in 2013 lies within this distribution, then there's no evidence for selection. And we're looking for SNPs where the observed frequency is actually outside the distribution. And we can calculate empirical p-values by counting up the number of simulations where the simulated frequency is more different from the median compared to the observed frequency. Most of our SNPs are fairly neutral. So here, again, the colored line shows the observed allele frequency in our population from year to year, and the black and gray lines show expected frequencies from our simulations. But we do find evidence of selection at one particular SNP. And this is total speculation right now, but if you look at these trajectories, you can see that the observed allele frequency starts deviating from our gene dropping expectations in 2008. In 2008, there was a high mortality event in our population in which 40% of our population died within a span of 2 months. We strongly suspect that this was due to disease. And so it's just curious that you see this deviation starting then. But we're doing a lot of follow-ups for this right now. Another thing you'll notice is that the allele frequency trajectories tend to bounce around through time. And we're currently working on ways to detect fluctuating selection in which we can compare the observed variance in allele frequencies through time to what we would expect from our gene dropping simulations. Finally, another fun thing we can do with this approach is to look at the expected genetic contribution of particular individuals to levels of population diversity, population genetic diversity in our population through time. So from the long-term demographic modeling, we know that there's really high variance in lifetime reproductive success. Here I'm showing you a histogram of lifetime reproductive success measured in terms of the total number of fledglings produced. Because we have the pedigree, we can actually pull out all of the descendants of a particular individual. So for example, this female born in 1985 has a lifetime reproductive success of 5. None of her offspring survived to produce offspring, so this is the pedigree of all of her descendants. Here's another female, also born in 1985, who was way more successful. And you can see that her pedigree of descendants is much larger. And with gene dropping, what we can do is generate the expected individual genetic contribution to levels of genetic diversity through time in our population. So these, for example, this one not super successful female would have contributed to the population in 1987 and 1988, and then is no longer going to contribute to our population. Whereas the more fit individual is expected to contribute a lot more to the population, and her her genetic contribution is also expected to last a longer time. One thing to note is that these simulations, these results I'm showing you here are done for single alleles, and so they're just expected contributions. In our future work, we're going to be identifying individual haplotypes and simulating transmission of specific haplotypes down the pedigree, which will allow us to actually calculate an individual's contribution to the population. And this approach will provide a lot of insights into the genetic basis of fitness in natural populations. So to sum up, today I've talked to you about gene dropping, which is a really powerful way of modeling neutral processes in populations with known pedigrees. We showed that there are many alleles in our population that do show large allele frequency shifts over just 23 years, although many of these trajectories are driven by gene flow, which is consistent with the high levels of immigration into our population. But even accounting for gene flow and drift, we can still detect rapid evolution of genotypes due to short-term selection. So none of the work I talked about today would be possible without the many, many students and interns at Archbold who collected all the data. I'd like to thank my collaborators, the folks who've given me comments on this work and my funding sources. And finally, I'm really excited to announce that I'll be starting my own lab at the University of Rochester next summer. And so if you're interested in thinking about population genetic inference with pedigrees or evolutionary genetics or conservation genomics in natural populations, please come talk to me. And with that, I'd like to thank you for your attention, and I think I have time for one question. Yes?
I think I missed this. What is roughly the census size of the population?
So